Hello guys again, this is Mohamad Al-Darazy again back with your favorite subject, biology. In this video I will be discussing document 5, synapses of chapter 9. This is the first video on the topic, so expect another post later on after we finish the things required in this video. So let me note first of all that I'll be using text and images from your own book, from your textbook, just so that you find it easier to relate when you're reading in the book at home. So let's kick things off with the introduction. We're going to read it together and try to understand as much as we can from this paragraph. They said, following an effective stimulation. So what does effective stimulation mean? You should remember and recall that stimulation is effective when it can generate an action potential, and it's ineffective when it is below the threshold and unable to do so. You can review document 3 for more details on that point. For now, let's continue. We said an effective stimulation leads to the nerve message being generated. Then it is capable of self-propagating along the neuron. Again, this is an idea that we discussed before in document 3 as well, how the action potential after being formed will jump along the neuron. If it's a myelinated fiber, it will jump from node to node. If not, it will have a continuous flow till the end of the fiber. So what about the problem we have here? Since the nervous system is made up of a network of interconnected neurons, meaning that neurons don't run on their own, they need to communicate with other cells to achieve their purpose. So as you can see in this image here, we have a network of several cells, of nerve cells connected together. How does the information circulate between these cells, between two neurons or between a neuron and another cell? We call an effector cell. An effector cell, by the way, is a cell that receives orders from neurons like a muscle cell or a gland cell. So how are cells connected with neurons and how are these connections organized? First of all, the book kicks off the activity with a definition of the synapse. They say that the synapse is a junction between the terminal bud of an axon of a neuron and another structure. This latter could be another neuron or an effector cell, as we said, like a muscle or a gland cell. So you can see in this image the three basic types of synapses. In all cases, we have a neuron connected or communicating with another cell. In the first case, the neuron is connected or communicating with another neuron, so we call it a neuron-neuronal synapse. The second case, you see that the neuron is connected to muscle cells, so we call it a neuromuscular synapse or neuromuscular junction. And in the third case, you see the neuron communicating with a gland or gland cells, so we call it a neuroglandular synapse. Now, if we go further into the details of a neuronal synapses, one neuron can be connected to another neuron in one of these different ways. Here you see the first neuron connected through its axon to the dendrites of the other, so we call it an axodendritic synapse. Here the axon of the first neuron connects to the soma or the cell body of the other neuron, so it's an axosomatic synapse. They can even connect an axon to an axon, or we have a connection between an axon and axon, and we call it an axonic synapse. And finally, we have a connection between dendrites of two neurons, so we call it dendrodendritic synapses. This is the same idea, but the image that they use in the book, so now you can relate these two ideas together. Now, they also said in the book that there are two types of neuronal synapses, an electric and a chemical synapse. So these two images present these two different types. The one on the right, this one, talks about electrical synapse. You can see here that these two cells, this is one neuron, this is the other neuron. There are connections between them called gap junction channels. These channels, as you can see, can allow the passage of what? Of ions back and forth between the two cells. So there's actual continuity between the two cells as if they're a single cell. This would allow the action potential to travel from the first cell to the other by electric mechanisms. However, in the book, they don't speak further of electric synapses. This is the only thing you need to know about them. We're going to focus more on chemical synapses, which are the ones you see here. The main difference you can see is that there is a space here in between the two cells. They're not actually in contact. There's a space, and you can see that these small red dots, these chemicals, are coming from one side to the other. So these are the chemicals that will facilitate the transmission, and thus this is why we call them chemical synapses. Let's look at the structural details of the synapse zone. First of all, this is a general view of a synapse. This is here the first neuron bringing the message. It's called the presynaptic neuron. The one receiving the message is the postsynaptic neuron, and this region here is the synapse zone. Now note that sometimes this cell is replaced by a muscle or a gland, but the idea is the same. If you look closely at the synapse zone, you can see that, again, this is the space in between called the synaptic cleft. These are the vesicles. These structures, the circular ones here, are called synaptic vesicles. You can see that they contain small dots. These are chemicals called neurotransmitters, or sometimes we call them neuromediators. And on the opposite side, on the other neuron, we have, this is the postsynaptic neuron, obviously, we have receptor proteins. And you'll find out their functions later on in this activity. Now again, let me repeat this is the presynaptic neuron and this is the postsynaptic neuron. Pre means before, post means after. You should expect in the exam or in exams to see images similar to this one. It's obviously very clear. So let me just help you to understand better what's happening here. This is an actual neuromuscular synapse. This is how it looks under the microscope. So sometimes they ask you to identify the different parts of this synapse. The trick is to look at particular details. Like first of all, the big division here, the big line, this usually represents the synaptic cleft, separating the two cells. And the small circles here, you see, which look like slices of pepperoni on a pepperoni pizza, these are the synaptic vesicles. So wherever we find these, we know that this is the presynaptic neuron. So then this would be the postsynaptic cell. In this case, it's a muscle cell because you can see the muscle fibers here running parallel to each other. And it has plenty of mitochondria because remember, muscle cells are very active cells that require plenty of energy, and mitochondria can facilitate production of this energy by glucose oxidation. Next, this is an image of a neuronal synapse. Again, it's not that easy to identify, but you can see here that this is the line running in between the cells. This is the synaptic cleft. If you are able to detect the vesicles, which are usually circular, these are the synaptic vesicles supposedly here. So this is the presynaptic neuron and this is the postsynaptic neuron. What next? Brace yourself. Homework is coming. So next I want you guys to open to pages 192, 193. We have these experiments here, 3 experiments. This is the setup, document C. I want you to interpret these experiments, and after you send me your answers, I'll send you my feedback and we'll discuss the answers in a different separate video. Have a nice day.